So on to the final session, the keynote talks. So first off, very happy to welcome Terence Risby. Terence is Professor of Environmental Health at Johns Hopkins Bloomberg School of Public Health. He's a member of the Acetone Scientific Advisory Board. He's a founding member of the International Association for Breath Research and the Journal of Breath Research. He's currently associate editor of the journal Biomarkers and the Journal of Breath Research. Terence has brought his expertise in chromatography and mass spectrometry to bear on applications relevant to research on a variety of diseases, including liver disease, and finding unique biomarkers in breath that relate to the functional status of the disease. The current focus of Terence's research is the use of breath biomarkers in clinical molecular epidemiological studies. Terence will also be joining us on the panel discussion tomorrow afternoon. So his talk is lipid peroxidation, a measure of human homeostasis. Thank you, Terence, for joining us. Over to you.
Thank you for the invitation to present a talk on lipid peroxidation and human homeostasis. I am presenting my talk today from a personal viewpoint, and I'm going to be using a lot of my information that has been generated by my laboratory. Let's start off and talk about oxidative stress and oxidative stress status so that everybody has the same status. Approximately 5% of the oxygen inhaled, which is required for respiration, is incompletely reduced to reactive oxygen species. A typical adult weighing about 75 kilos requires, for normal respiration, this is when they're breathing tidally, about 20 moles of oxygen a day. And therefore, each person will be exposed endogenously to reactive oxygen stress of about 1 mole of oxygen per day. Reactive oxygen species can damage cells, tissues, and organs. Therefore, to protect against ROS, all aerobic cells contain antioxidant systems. Oxidative stress status is the balance between oxidative stress and antioxidant defenses. Oxidative stress has been implicated in the initiation or the exacerbation of specific diseases or dysfunctions, and is also involved in aging. This is why, as you get older, one of the things that happens is the antioxidant defenses decreases. Reactive oxygen species are involved in molecular signaling, and they are involved because they are easily reversed by antioxidants. Reactive oxygen species are involved in host defenses, inflammation, and in the phagocytosis of foreign species in the lung, including airborne particles. What does ROS do to cells? They can damage DNA or nuclear bases, which leads to mutagenesis and carcinogenesis. They can damage nucleic acids, which can lead to mitochondrial dysfunction. They can damage proteins and modify normal function of enzymes, receptors, and transporters. They can damage polysaccharides that could lead to connective tissues disorders and musculoskeletal disorders. And finally, they can damage to cellular lipids that can lead to membrane leakage and cell damage. What are cellular antioxidant defenses? Generally, they are enzymes such as superoxide dismutase, catalase, glutathione peroxidase, heme oxygenases, etc. These are inherently in all cells, and then there are exogenous or endogenous non-enzymatic species such as vitamin A, vitamin E, bilirubin, glutathione, urate, ferritin, and isoprene, which is produced by the biosynthesis of cholesterol. Isoprene is an endogenous antioxidant. In terms of oxidative stress and breath, only the products of lipid peroxidation can be measured in breath. And the relationship between VOCs in breath and oxidative stress has been studied for more than 50 years. Probably the earliest, most definitive study was published in Science in 1974 by Riley, Cohen, and Lieberman. In this study, mice were injected with carbon tetrachloride, which is metabolized by the liver to produce free radicals that generate the production of reactive oxygen species, such as superoxide, hydrogen peroxide, and the hydroxy radical. This in vivo study showed that lipid peroxidation is initiated by reactive oxygen species, and simple alkanes, ethane and ethylene, were detected in the gas of the chamber containing the mice. The most critical aspect of this study was that these researchers showed in vivo that the relationship between carbon tetrachloride and alkane production could be blocked by prior injection of the antioxidant vitamin E. This study demonstrated that the relationship between reactive oxygen species and lipid peroxidation, and the fact that lipid peroxidation could be blocked by antioxidants. This is the basic schema of lipid peroxidation. The first step, based on hydroxy radical, is the abstraction by the hydroxy radical of an allylic hydrogen to produce water. The remaining radical conjugates and then is peroxidized, and depending whether we have an N3 or N equals 1 or N is greater than 1, you end up with producing hydrocarbons such as ethane, aldehydes such as malonyl dialdehyde, and conjugated dienes. This is a chain reaction that occurs all the time and in all systems. The products of lipid peroxidation are very dependent upon the identity of the fatty acid. They can be straight and branched chain hydrocarbons, conjugated dienes, lipid peroxides, aldehydes, ketones, fatty acids, and isoprostanes. A lot of these molecules can be found in breath, depending upon their volatility and stability. Breath biomarkers of lipid peroxidation can be used to follow therapeutic interventions or pharmacologic interventions, and lipid peroxidation will occur acutely or chronically. And examples of their potential uses will be presented in the following discussion. Acute markers in breath are hydrocarbons, since these non-polar chemically inert VOCs will cross the cell membranes immediately and will be transported to the lungs in the blood and excreted in the breath. Chronic markers in breath will be directly related to the vapor pressures of the products, their polarities and stabilities. Let me talk about the first application of lipid peroxidation that we did in our laboratory, and that was to look at acute reactive oxygen stress injury in a specific organ, and we used as a model reperfusion injury during organ transplantation. During organ transplantation, when the donor liver or a donor organ is collected from a recipient, the ATP is degraded to hyperxanthine during ischemia. Also, the enzyme xanthine dehydrogenase is converted to xanthine oxidase. And after a period of ischemia, the enzyme xanthine oxidase has its substrate, which is hyperxanthine, present in the tissue. And so that enzyme can convert hyperxanthine to xanthine, and then on to xanthine, and then on to urate and the production of superoxide. And this occurs when blood is restored to the ischemic tissue, and that is known as reperfusion injury. This slide is a little busy. It is the first human liver transplant that we studied at Hopkins. And what I'm showing on the y-axis is the amount of ethane is exhaled, and we're normalizing it to minutes body surface area. And the x-axis is the time of surgery. And there are basically three parts to the system: a baseline where all the initial dissection occurs. The anhepatic phase is when the liver is removed from the recipient. And finally, reperfusion, when all the connections to the liver are made. And the first process is the portal vein is connected and is opened, and that supplies approximately 75% of the oxygenated blood. Subsequently, the hepatic artery is opened. The injury at transplantation is determined by the time and temperature of the ischemic tissue for the donor liver and the corresponding injury when the blood is returned to the new liver in the recipient. What I'm showing in the black squares is the measure of ethane during the surgical procedure. And you should notice this surgery took approximately 12 hours. And it took about 5 hours to get the liver out. And then first anastomosis for the portal vein is approximately a little over 80 minutes, 90 minutes. And the first thing you should notice is until the portal vein, we're around 2 or 3 nanomoles of ethane in the breath. And then when the portal vein is open, we have a huge increase in ethane. And this actually represents the first time that during transplantation, reperfusion was measured independently of ischemia. This is the summary of the next 15 liver transplant patients that happened at Hopkins, and I was collecting breath from each of these. And what you should notice, we're measuring, once again, exhaled ethane in nanomoles per body surface area per minute, and you should notice that the patient goes in with about 4 nanomoles of ethane. It drops a little bit during an hepatic phase, and it increases at least twofold when 1 to 3 minutes after the portal vein is opened. And there is no second increase when the hepatic artery is opened. This was showing that reperfusion could be measured without any impact on the normal surgical procedure. This is a different model, and here we're looking in an animal model, a model where the coronary artery is occluded for a period of 2 hours of warm ischemia. And then just before the occluded artery was opened, the animal either got SOD mimetic or was a control. And the blue is the control. And you can see that 5 to 8 minutes post opening of the reflow of the coronary artery, you saw an increase in ethane, which is reperfusion. And that reperfusion was blocked if the SOD mimetic was given prior to opening the coronary artery. And clearly, this shows that you can block reperfusion injury by adding an antioxidant just prior to reflowing blood into the transplanted organ. And this represents a significant benefit to organ transplantation, because if you can reduce reperfusion injury, you can improve outcome of the transplanted organ. There are many other applications of acute interventions. I'm not going to present them today, but if you look in my biography, you can find evidence of looking at radiation, etc. The critical requirement of any of these studies is the collection of a reproducible representative breath sample. The studies that I talked about during surgery, each of those patients were intubated, whereas now the study subjects are breathing normally, and so you need to collect breath from them. And as soon as you ask a person to breathe for you, they tend to hyperventilate. Men are much worse than women. What I'm going to present today is talking about reactive oxygen species injury and disease, reactive oxygen injury and diet, reactive oxygen injury and exposure to toxic agents, and reactive oxygen injury and pregnancy, and finally looking at exercise. This is the first study that we did using people who were breathing normally, and it was done in children whose ages were between 1 and 17 years old. We had 8 patients with severe chronic liver disease, and as a result of the liver disease, had vitamin E deficiency. And you can see that since they have vitamin E deficiency, they're oxidatively stressed. And you can see they have a large level of ethane. And this time we're monitoring ethane in terms of picomole per kilo per minute. For each of these children, we had controls, which they were their siblings, or age-matched healthy controls. And we had 9 of those. And what you should notice is post-vitamin E therapy, the level of oxidative stress in these patients returned to what was the same as the control fasted SIBs or non-fasted controls who were not siblings. And clearly, you can follow vitamin E deficiency in children. The alternate way to follow vitamin E deficiency is to take a fat biopsy. And I assure you, if you're trying to deal with children, they much rather give you a breath sample than give you a fat biopsy. This is a study that we did at Hopkins where we studied approximately 45 women during their pregnancy for their entire pregnancy when they came to the OB clinic, the obstetrics clinic, on a monthly basis. And we followed their reactive oxygen status, oxygen status during the pregnancy. Unfortunately for this study, the normal protocol in obstetrics is to provide mega doses of vitamins, antioxidant vitamins, to mothers during pregnancy. And so we were unable to observe changes in oxidative stress during pregnancy. However, we were able to identify which one of the mothers was smoking. And you can clearly see that smokers had approximately twice the amount of ethane in their breath, which is indicative of their oxidative stress as a result of cigarette smoking. Remember, typically these mothers, when they come to clinic, probably haven't smoked for at least an hour, probably 2, because they wait a lot of time in the clinic. So, but clearly you can identify that there were 26 non-smokers and 19 smokers. Also interested in this study, we took breath samples from their neonates 1 day after delivery. And we compared the neonates of the smoking and non-smoking mothers. And also, there were 2 groups of mothers that fed their babies formula and mothers that breastfed. Remember, breastfeeding during the first few days after delivery, the breast milk is rich in lactoferrin, which is a really strong antioxidant. And what you should notice is the breath of the 1-day-old neonate is significantly elevated in the smokers versus the non-smokers. The error associated with the smokers breastfed neonates is because there's only 2 mothers breastfed who were smokers. And clearly you can see the benefit of non-smoking breastfed, the breath of the neonate of non-smoking mothers who were breastfeeding them. And it's the least oxidatively stressed. This is a study that we conducted of the DASH diet. The DASH diet is a method of controlling hypertension using diet. And for this study, we had 123 subjects. These were older than 23 years old. And the requirement was that the person came to clinic every day of the week for morning, lunch, or dinner to eat 1 meal and then collect food for the other 2 meals and go back. And they came for 3 weeks, and everybody started by eating the B diet, which is a typical American diet, which is low in fruit and vegetables and high in fat. After a 3-week period, the study subjects were split into 3 groups. The A diet, which is the DASH diet, the B diet, they stayed on the typical American diet, or the C diet, which is the same as the American diet, but 5 times the amount of fruit and vegetables. Before they switched their diet, we collected breath samples. And after 8 weeks of being on the second diet, we collected another sample. This required an awful lot from these people to come every day to eat a meal and collect food. And these, by the way, are not students. They are, most of them were in their 40s and 50s. These are the diets. The B diet is the typical American diet, high in fat, low in fruit and vegetables. The C diet is the same amount of fat, but 5 times the amount of fruit and vegetables. And the A diet is the DASH diet. And you can see it's low in fat and 5 times the amount of fruit and vegetables. These are the diets. These diets were made up by the USDA, and we were not aware of which subject was in which group. What I'm showing here is looking at each individual where they are his or her own control, and we're looking at the change in breath ethane from baseline to the end of the study. And what you should notice is if you're on the typical American diet, you're becoming more oxidatively stressed as a result of the second 8 weeks. Whereas the DASH diet, you can see you're becoming less oxidatively stressed. And the diet C is a little worse than the DASH diet. And clearly, the only intervention in these people is to be on this controlled diet for 11 weeks of study. Let's look at some effects of exercise on exhaled breath. And for this study, we had 14 healthy adults greater than 40 years old. And the protocol was controlled bicycle exercise with different resistance or workloads. And what happened was the study subject pedaled at a given speed for 5 minutes, and then we increased the workload. We went from 24 watts per second all the way up to 150 watts per second. We were monitoring their blood pressure, and they could stop the exercise at any time. We collected breath for 1 minute after each workload increase. And we monitored the ventilation, which is the minute ventilation, the oxygen consumption, the CO2 production, the anaerobic or lactate threshold, and we monitored that continuously. In addition, we injected technetium into this subject, and we had them sit with their chest continuously in a gamma camera, and we measured cardiac function, cardiac output, heart rate, stroke volumes, blood pressure, peak filling, and peak emptying rates continuously during the exercise. This slides shows the cardiac output and minute ventilation as a function of exercise or workload. And you can clearly see the red is the minute ventilation and the blue is the cardiac output. What you should notice is the cardiac output increases much less than does the minute ventilation, which is obvious. If you think about it, is what are you doing when you're breathing? The blood goes to the lung, and as you increase the blood to the lung and you increase the ventilation, the concentration of breath changes. What I'm showing here is the normalized breath acetone as a function of exercise or workload. And here, instead of talking about normalizing the breath to body weight or per minute, what we're normalizing to physiology, which is the amount of carbon dioxide being generated as a result of breathing. 14 subjects were able to exercise to 100 watts. Only 9 subjects went to 24 watts, 124 watts per second, and only 3 reached 150 watts per second. And this is why, as you look at the later ones, 120 watts, you have more of a standard deviation of the error. But what you should notice is during the initial part of exercise, the acetone drops and then starts to increase. And what is happening here is the person is changing his energy source to burn fat. This is the same and a different marker, and this time it's breath isoprene. And remember, isoprene is produced during cholesterol biosynthesis and is produced in the liver. And what you clearly see is blood level of isoprene decrease as a result of increased ventilation in the lung, and the amount of isoprene is blown off from the lung during the exercise. So you can see that isoprene, blood levels of isoprene drops as a result of exercise. If you look at the same subject later on that day, the level of isoprene in breath has increased. This is looking at oxidative stress, once again looking at breath assaying as a function of exercise. And what you should notice is there essentially is no change in breath ethylene as a function of exercise. The only thing you're doing is increasing the amount of oxygen used. You're not decoupling the antioxidants during exercise. So exercise is good for you. It doesn't increase oxidative stress. What I'd like to talk about now is to look at reactive oxygen species, aging, and obesity. One of the biggest problems with the US and probably a lot of Europe is that as you age, you become obese. And one of the things that goes with obesity is disease such as diabetes and cancer. And the question was, is that related to the decrease in oxidative stress? And so what we're doing here is a lifetime diet manipulation study where we took Fisher 344 female rats. We had N equals 22, and we fed them either ad lib for 24 months, or we diet restricted them 30% less than ad lib. And so at the end of 24 months, the ad lib rat weighed about 290 grams, and the diet restricted rat only weighed 170. So obviously the fat and sassy rat is almost double the weight of the diet restricted rat. And so what we were looking at is we measured the breath ethane in these rats after 24 months. And the first way we did was we normalized it to body weight. But if you think about it, if you normalize the ethane to body weight, essentially you overcorrect the fat and sassy ad lib fed rat. So therefore, when we normalize to respiration, in other words, to the amount of CO2 production, you clearly see that the ad lib rat is much more oxidatively stressed than the diet restricted rat, and it is statistically significant. My experience from breath research is, and my view of the future of breath research is, the most successful future breath tests will be used to monitor therapeutic and pharmacologic interventions. Why? Because each person is his or her own control. In other words, you take a person, you give them an intervention, and you can see the change in breath as a function of the therapy. And I think exciting areas are the efficacy of pharmacological interventions, such as immunotherapy for cancer. Following the status of transplanted organs, you can do this with initial function or rejection. Rejection involves inflammatory events, or identifying infections before symptoms are apparent. Here, what we're not looking at the identity of the infection, what we're looking at is the host response to the infection. And this is totally generic to any infection. It's much like taking a temperature of a person. If you don't want to do research where you're looking at intervention for an individual and you're looking at populations, the careful selection of control subjects will define the utility of any diagnostic breath test. Comparisons of breath concentrations with different subjects, you should ideally compare breath markers as a function of respiration. You need to be very careful when you collect breath from individuals. You need to know what they did in terms of diet, their exercise, what prescription drugs they're taking, because these factors could have a major effect on the breath. The second thing that you need to really be very careful of is room air. Room air in clinics is typically pretty bad because, there's a lot of things going on. And now, of course, with infections, everybody is using alcohol to wipe their hands. And so the air in clinical labs is rich with ethanol. Another thing that I think is critical is breath research is a group effort involving very different disciplines, and each group of people play a major role in defining breath tests. And finally, my view, breath analysis is only good as the method of sampling. And the method of sampling, you need the person to relax and breathe normally. By the way, women give much better breath samples than do males. Let me conclude by acknowledging my collaborators, but first of all, let me acknowledge patients and controls of all the people that I've recruited for my studies. I'd like to acknowledge my former graduate students and fellows, clinical fellows, acknowledge my clinical colleagues in various disciplines, surgery, anesthesiology, cardiology, and pulmonology. And I acknowledge clinical colleagues at other institutions in London, Holland, and in the US. And finally, I acknowledge clinical and engineering colleagues at other institutions who have helped me build instruments designed for use for breath analysis, and support from the National Institutes of Health, the Air Force Office of Scientific Research, and the National Science Foundation. Thank you for your attention.
Fantastic. Thank you so much for that. Really appreciate it. We do have a few questions. I was actually going to celebrate the end of the conference tonight with a cheeseburger, but now I'm worried about what my breath ethane levels will do.
You're not fat and sassy.
Yeah. One question I had. So it seems that obviously that compound is very well validated, and there's a lot of evidence that are suggesting that it does relate to oxidative stress. Interested in your perspective about why it's not translated through into a clinically deployed test, given that it obviously is pretty robust.
Well, in terms of reperfusion, we did do studies where we used the superoxide mimetic in kidney transplants, where you had a donor received, the donor has 2 kidneys, and then you put it into recipients where one recipient received normal reperfusion, and the other one we put in the SOD mimetic and blocked reperfusion, and there was a better outcome as a result of that surgery. We looked at the same thing with radiation, doing total body irradiation and looking at effective radiation, and you can see the benefit of oxidators, giving antioxidants. In terms of, the first example with the kids that were on the list for a liver transplant, and as a result of having severe liver disease, we showed that you could give them vitamin E, and they would be feel better while they were waiting for the transplant. So that's a therapeutic, and this is a result of giving a breath test. Why didn't it catch on? It's a good question. This is, one, we were an academic environment where we were looking at clinical. We were not generating, the goal of the research was not to build instruments. That's the goal of others.
Yeah.
We did do some studies with an instrument that was built in the Netherlands by SensorSense, and you can look at, you can use them during cardiac surgery in real time. The problem with the reperfusion is that at the time, we were not doing real-time analysis. In other words, we weren't doing something where you actually could get data while the surgery was going on. We did do subsequently, when we had an instrument that was able to measure oxidative stress in real time. You could, in fact, do interventions. We didn't do the interventions, but we show in real time during a surgical procedure. And these studies were done at the Harefield Hospital in London. So yeah, you can do things in real time. We did it, but we didn't use it to actually tailor-make the surgical procedure.
That's great. Thanks so much. So one question that's come through is, great summary, thinking of how to use reactive species for oncology therapeutics. Can we accurately and predictably enhance local oxidation, e.g. enhancing photodynamic therapy with sensitizers, but limit systemic toxicity? And also how to continuously monitor the amount of light activation during treatment?
That's a really interesting question. I think if you're interested in looking at oxidative stress in real time and an intervention, you have to use something that's going to happen in real time, which limits you to ethane measurements or ethylene measurements rather than aldehydes or ketones, because they take a lot longer to be excreted in the lung, and it's totally doable.
Interesting.
When we actually did try doing ethane in real time using ICOS, and we were able to do it, but the instruments are a bit big. It's a sort of a boat anchor, and you have to take the person to the instrument rather than the instrument to the person. Okay. But that was because, you know, technology is changing. If you can build smaller devices that you can measure hydrocarbons relatively quickly and easily, and you can potentially take them to the clinic and actually measure things in real time when you're looking at intervention.
Fantastic. That actually, you touched on another question that I had. So obviously you're talking a lot about ethane, and you mentioned aldehydes. That's something that we've seen published in literature. We're interested in it as well. But it sounds like what you're saying, that ethane has an advantage in the sense that it is excreted into the breath much faster than some of the aldehydes. Did I get that right?
Correct. You are absolutely correct. Also, aldehydes are very easy to oxidize. And so that's, you know, hydrocarbons are essentially neutral. They're non-polar. They come out of they go in the breath. They're pretty stable. And that's why isoprene is such an effective antioxidant, because it's a hydrocarbon. And the only thing that's there is some regions of unsaturation.
Interesting. So one other question that's come in. So you mentioned where people are their own controls. That as well to me makes a lot of sense. So the question here is, that being the case, should we develop a profile of healthy people? So like a 23andMe of healthy VOCs.
I would have said that to be absolutely true, but I think COVID-19 has in fact dealt a whammy to everybody, because there are many people who are asymptomatic but are in fact infected with COVID-19, and if they are infected, their host responses are different. So I would have, if prior to COVID-19, I would have said, yeah, that's easy. Now, until we have a vaccine, we don't know if a person is potentially infected. And therefore, if you are infected, your host responses have everything has changed, and your breath profile is. So what I'm trying to say is, using each person as his or her own control is the safest way of looking at an intervention. Okay.
And that actually is probably going to be covered in tomorrow's session, but from what you're saying there, it does sound like that host response would potentially be something to look at from a diagnostic standpoint in COVID. Is that, yeah. Okay, time for a couple of other quick questions. One is, you know, if you were to do any of your work over again, would you do anything differently or change the approach in any way?
I think honestly, every mistake I've made in the process of doing this research. I started this research in the 80s, and we used breath just because in the 80s there was the HIV infection and everybody was afraid to look at urine or blood because they were afraid of getting infected with actually, and now we have come the complete circle because now you can be infected by doing breath research. So I made every mistake. The advantage that we did do was we did looked at surgery where the patient is essentially asleep and being breathed by a machine. But every other mistake I've made over the years, with hindsight, I would have done a lot more studies if I hadn't made every mistake that's going. Yeah, I mean, sure, there are lots of things we could have done, but it's a fact of life. We've done it.
Indeed, indeed. So last question, any big predictions for where breath will be in 5 years' time? You know, either technology-wise or a kind of killer application out there? I
honestly believe. Well, let me say, everybody. If you look at the studies that have done, and we've tried doing studies on lung cancer, and the problem is people do not tell you if they're smokers, and it is such a shame to go do studies and then find out all the data that you got was just completely destroyed by the smokers. Because if you think about it, I always joke that smoking cigarettes is equivalent to sucking on the tailpipe of a truck. Almost every compound you can think of is in cigarette smoke. But you know, we've done studies looking at lung cancer and cancer, and cigarette smokers really confound the data. And I think it's shown by the neonates, the study that we did where we looked at pregnant women and their neonates. Here are kids where the mother hasn't smoked for maybe at least probably 2 days, and the neonate is still getting effects of smoking from its maternal. So you can see that smoking has such a huge effect on breath. Where do I go? I think that the comment I made about intervention, whether it's pharmacologic or surgical intervention, you can look at this in real. You can see the effect of intervention, and I think this is the way to go for the future right now.
Brilliant, excellent. Thank you so much. With that, we will move on. Really appreciate it, Terence, and we'll see you again tomorrow for the panel discussion. So thank you.
Thank you for the invitation.